He was the obvious man to act as a roving investigator on viral diseases for the US Armed Forces, and he went on to do the work on dengue, sandfly fever, and Japanese encephalitis that Walter Schlesinger has just told us about. In contrast, at that time, in 1940, I was a raw medical graduate and served first in a field ambulance and then a casualty clearing station in Palestine and Syria before I was taken on as a pathologist at the hospital and then as a malariologist, where I worked in the Southwest Pacific area for the rest of the war. Not a career that offered any chance to do laboratory research. However, I was lucky that as a malariologist, my immediate superior was Colonel Keogh, a man who'd worked with Burnett at the Walter and Eliza Hall Institute in the mid-1930s, and he was able to persuade Burnett to offer me a job at the Hall Institute when I was discharged from the Army early in 1946. Coming new to virology and able to embark on something more productive than military service, as it was to me, I read extensively. One of the journals that I read most assiduously was the Journal of Experimental Medicine, at that time a major journal for virological papers. And of course I came across the Savin, Ilitsky papers of the late 1930s, which Igor has spoken about. Then in 1948, when Burnett asked me to collaborate with him in the production of a little book called The Production of Antibodies, I well remember preparing for this by reading, amongst many others, a series of 4 papers in the British Journal of Experimental Pathology on the mechanism of immunity to filterable viruses, part of the product of Albert's year at the Lister Institute in 1934. Thus, I was acquainted with Sabin as the name of a person whom I had to meet when I came to the Rockefeller Institute to spend what was essentially a postdoctoral year with Ronnie DuBose in 1948-49. Albert had then moved to Cincinnati, but I met him at a symposium on viral diseases held at the New York Academy of Medicine in December of 1948. In fact, my wife met him first, for she was sitting next to him in the audience when I was presenting a paper on the pathogenesis of the viral exanthemeter. As was the habit of people brought up in Australia and England, I was pronouncing titer as tita, and Sabin asked the young lady sitting next to him who this young man was. She said rather defensively, "My husband." And I met Albert again at the 1949 ASM annual meeting when he spoke about pleuropneumonia-like organisms, as mycoplasmas were then called, as well as commenting in a critical and incisive manner on presentations on virology. From down under, there was a dramatic contrast between the national scene in virology in the United States at this time and the situation in Australia. Virtually the only virus research in Australia in the immediate post-war period was that being done in Burnett's laboratory. It was a lively group, not only because of Burnett himself, but because he'd gathered some interesting people around him: Beveridge, Gottschalk, Cairns, Fazekas, French, and others. However, it was small and it was very isolated, for no one visited Melbourne then unless he came for a prolonged stay. It was on the way to nowhere except Antarctica. Here in the United States, the situation was very different and very stimulating. There were a multiplicity of centres working on all the then known viruses, except perhaps my own favourite, ectromelia virus. From Australia, the Northern Hemisphere was rather inaccessible. One did not take a 4-week-long sea voyage and a 4-week-long return trip for a few days or weeks in Europe or the United States. At that time, PhD degrees were not awarded in Australian universities, and at that time also Australia was looked to England rather than the United States for doctoral training and postdoctoral training. This isolation was soon to change when the intercontinental air routes were opened up. And from 1953 until 1967, the period when I was actively engaged at the bench, I used every few years to make a 5-month-long trip around the world and visit virtually every centre of virology in the United States and Europe on the way. Later, with jet travel and involvement in international science, the number of such trips rose to as many as 6 or 7 a year. But I would remind you, and I'd remind you also that Albert has undertaken similar travelling experiences. It's a gruelling trip from Australia, over 20 hours flying time across some 10 time zones every time you want to come to Washington, London or Geneva. During these overseas trips, I've had the pleasure of meeting Albert on many occasions, both on his home ground in Cincinnati, as well as conferences in Cold Spring Harbor and elsewhere, and twice in Israel when he was president of the Weizmann Institute. Once in 1973, we shared the hospitality of the NIH as fellow faculty scholars. And then during the last few years, my wife and I have enjoyed evenings with Albert and Eloise at their apartment in Washington and at Bob Channock's home in Bethesda. It wasn't until his 70th birthday, about in 1976, that we were able to welcome them in Canberra, and Ian Gust was able to have a 70th birthday celebration, at which I'm told Albert danced all night long in Melbourne. What then are my impressions of Albert Sabin as a scientist, speaking now as a scientist who knows something of the American scene but is not part of it? Clearly, his name will be forever associated with the activities in developing oral polio vaccines and establishing their worldwide use. However, I would remind you that he was elected to the National Academy of Sciences at a time when there were few medical scientists in that academy in 1951, before the work on polio vaccines had begun. I'll therefore take a broader view and look at the totality of Albert's contributions to science. A striking feature of it, which is attributable partly to the man but also to the time, is the great diversity of agents and subjects that he worked on. You already had a taste of that in the preceding papers. Other great microbiologists who began work in the 1920s showed a similar Catholic interest in infectious diseases. I need only mention Christopher Andrews and Macfarlane Burnett. Albert had published several papers on pneumococci even before he had graduated in medicine. And then, although primarily a virologist, he carried out classical and extensive studies on toxoplasmosis and mycoplasmosis over many years. His experimental work on poliovirus dates back to 1931. Work on herpes virus extends from the discovery and identification of B virus to studies of their role in tumorigenesis. And an abiding interest in viral diseases of the central nervous system is dominated by studies on the pathogenesis of the viral encephalitides. I'll say little about poliomyelitis since many aspects of this topic will be discussed by other speakers, but it may be appropriate if I comment on some aspects of poliomyelitis in Australia which are relevant. In the late 1930s, I'd remind you, poliomyelitis was as dreaded a disease in Australia as it was in the United States. And we had a very large epidemic there in 1937, 1938. I was then a fifth-year medical student, and I spent my long vacation working as a volunteer medical officer at a local infectious disease hospital in Adelaide in an attempt to cope with the emergency. In Melbourne, Burnett was asked to undertake research on poliomyelitis because of the epidemic. It was very difficult to get monkeys for research purposes in Australia, and then there was no other way to work with the virus. He isolated the local strain of poliovirus in rhesus monkeys, and then the supply of these animals ran out. A few cynomolgus monkeys were shipped down from Singapore, and unlike rhesus monkeys, they proved to be susceptible not only after intracerebral inoculation, but also after feeding. As you'll remember, at that time it was widely believed that poliovirus entered the CNS via the olfactory bulbs for the reasons that Igor Tam has discussed. Intrigued by the results of the feeding experiments, Burnett did some surgery, placed virus on the intestinal epithelium, and found that not only were the monkeys infected, but virus could be isolated from the local lymph nodes. A few years later, Albert elaborated on the multiplication of poliovirus in the intestinal tract in his Bieler-Schick lecture, but the concept that poliovirus was primarily an enterovirus was not properly integrated into science until the classic paper in the journal Science in 1956. The other contribution from Australia to poliovirus was, like the early experiments of Sabin and Olitsky on the growth of poliovirus in tissue culture in 1936, a near miss. In November 1938, which was for Australia just after the outbreak of war, Burnett and Lush tried to isolate the local strain of poliovirus in cells obtained from the throat, intestine and lungs of a human embryo. They showed by inoculation of monkeys that the virus had multiplied in cells obtained from the throat and the intestine, but not in those from the fetal lung. Then, once again, the supply of monkeys ran out, and this time no more could be obtained since Singapore was in Japanese hands, and the work stopped. To return to the United States and to Albert Sabin. Wartime activities provided for him the opportunity to isolate the dengue and sandfly fever viruses and to become the world authority on these viruses, and to study methods of vaccinating personnel exposed to infection with dengue, sandfly fever and Japanese encephalitis viruses. Then, with the realization that poliovirus was indeed an enterovirus, came a group of papers on this group as a whole, including the recognition and naming of reoviruses as a separate family. Along with this work on a range of specific viruses, Albert Sabin has had a long-standing interest in several general phenomena, and Walter Schlesinger has outlined some of these. The effects of genetic constitution and of nutrition on susceptibility to viral diseases, the role of viruses in tumorigenesis, the vaccination against various diseases, both with inactivated virus vaccines and with live vaccines given orally, or, as in his most recent work, by aerosol. Thus, we can survey almost 60 years of publications in microbiology, a record remarkable not just for its volume, but for its quality. All Sabin's published work is marked by the careful selection of a problem, for after all, science is the art of the soluble, the application of the appropriate experimental procedures, and the presentation of a definitive result clearly enunciated. Albert Sabin has made many contributions to science, but he was above all a medical scientist. He was always concerned with human suffering and with how to present disease, not just understanding it scientifically. It was this concern with human suffering that led to his emergence from the laboratory to become an activist in support of oral polio vaccines and to work with public health officials and politicians to get them tested and used. You'll remember that in 1957 there was a good vaccine against poliomyelitis, the Salk inactivated vaccine, which was in wide use in the industrialized countries. Use of this vaccine had already greatly reduced the incidence of poliomyelitis in the United States, Australia, and in several countries in Europe. Many influential virologists said, why change from something that's proved its worth? But Albert Sabin was convinced that there were features of his live polio vaccines that provided such great advantages over a killed vaccine that they should be subjected to proper, large-scale field tests. Firstly, because they provided gut immunity, they would protect against infection with wild polioviruses and not just against paralysis. Secondly, since they multiplied and were excreted, they could infect and provide protection to contacts and not just to vaccinees, a very valuable ripple effect. And most important of all, the cheapness and simplicity of oral administration opened up the use the possibility of the use of polio vaccines for the great mass of the world's poor, for whom the cost of the injected vaccine was prohibitive. So Albert became a publicist and an activist and went out to convince the health officials of many countries and of the World Health Organization that his vaccines had to be given proper trials. I'm glad to say that it was a scientist from down under, once again Macfarlane Burnett, who helped him to some extent in this task, for Burnett was chairman of a critical WHO meeting at that time, which recommended that trials of oral polio vaccine should be carried out in increasingly large numbers of people in different countries. So that from my perspective, perhaps as a scientist from down under, or perhaps just as another virologist, I see Albert Sabin as one of the great medical scientists of this century, a man who investigated and illuminated a dozen fields in virology, as a man who had made his mark in science before he developed the poliovirus vaccines that have proved such a boon to mankind, and finally as a man who went out and got things done in the hard, practical world, if that was necessary to relieve human suffering. Thank you, Albert. Thank you, gentlemen.
Thank you very much, Frank, and let me thank the participants of this first session. We now have a coffee break, and we'll return in approximately 15 to 20 minutes, and your chairman for the next session will be Dr. Sheldon Wolf. Coffee break. It does work, doesn't it?
Okay, I just said it.
Just speak louder.
Yeah. Could we begin, please? Well, I shouted twice.
May I have your attention, please? We're ready to begin the next session. Shelley, it's all yours.
Thanks. I'll be here for the dinner tonight. Yeah. Will you be there? Good.
I don't know if there's anything to talk about, but we might as well.
Okay, good. Could we begin the second session, please? We are running a bit behind. Let me first make one announcement. Professor Walter Hennison, unfortunately, will not be able to join us today. Apparently, his passport was stolen in the airport. Dr. Horstman will move up to follow Dr. John Fox, and then Dr. Chanik will end this morning's session instead of speaking this afternoon. The first speaker on this session is well known to all of you, Dr. Frederick Robbins, who is presently at Case Western Reserve University School of Medicine, formerly president of the Institute of Medicine, and before that the dean at Case Western Reserve, will talk on the understanding and control of poliomyelitis. Fred?
Thank you, Dr. Wolf. Dr. Fauci. Craig Wallace, he isn't here, but I'll greet him anyway. Albert and Eloise. And many friends in this audience that we've met around these various topics so many times. And I, like everybody else, have to say how gratified I am to be a part of this particular meeting. This ought to be Albert Sabin year in the United States. He's gotten every honor there is to get, practically, and is getting more. But somehow I have a feeling that a gathering of his peers and colleagues, where none of us is peer, you understand, but his scientific colleagues perhaps means as much to him as sitting next to the president at lunch or dinner or whatever it was. By the way, if you have any messages to send to Reagan, and I might say I have a few. Well, after hearing the presentations that have preceded mine, I have just decided not to give what I was going to give. That's a habit I have. I prepare something, although in this instance, to some extent, I'm pretending I prepared something, and then change it in midstream. But it seems to me that many of the things that I was going to talk about have been covered or are going to be covered by people who know more about the topic than I. There's no question that what Albert is best known for is his work on polio. But as you've heard, it is not the only thing he's done that's truly outstanding. You've also already heard, to a certain extent, that he's been involved in almost every aspect of the problem of polio. He's worked on the pathogenesis. He's worked on the agent itself. He's worked on the immunology. He's worked on the prevention. And just as he's covered a broad span of microbiology, he's covered a broad span within the field of polio itself. His contribution to our understanding of the pathogenesis has already been mentioned, and the work that he did along with Robbie Ward has, along with work from the laboratories at Hopkins, Bday and Howe, and others, established quite firmly the enteric nature of the polio infection, which was of great importance in approaching its control. I think his contribution to the understanding and debunking, to some extent, of that rather unfortunate episode when we were putting sclerosing substances into people's noses is an important contribution which not everybody knows about. But he has, as everybody has mentioned, an instinct for what's important. And in 1936, he went for the jugular and is the first person to have cultivated the poliovirus in tissue culture. Now, he didn't do it quite the way, he didn't complete the job, but as Igor Tam stated, this was because he was using the wrong virus. I was unaware, Frank, of what Frank Fenner mentioned of the work that Burnett had done with tissue culture, and I'll have to learn more about that later. But there is no question that Albert Sabin was number one in cultivating the virus in tissue culture. And for me to say that is quite an admission, don't you think? But what has really made the difference was his painstaking work, along with Hennison primarily. It's too bad that he won't be here, of working out the attenuated strains, testing them systematically, and developing the attenuated vaccine. This was not easy to do in the United States because, as was already mentioned, we had an effective vaccine that was in general use. And one of the most important things that Albert has done in his career, in my humble opinion, is his work with the Russians. That relationship, which he was able to establish, and we'll hear more about later, seems to me illustrated something which is not totally unique, but is the potential role of science in establishing relations between people under conditions somewhat freed of the political and other constraints. And if that can be extended, that would be your greatest contribution, in my honest opinion, Albert. Now, where are we today in regard to polio? Well, in much of the world, polio is under control. And when I say control, I mean it isn't a big problem. That's different from eradication. But it isn't even under control in a lot of parts of the world. And as was already mentioned, here is where Albert has been playing an extraordinarily important role. He has been the principal proponent of vaccination and control of polio in the developing world. And he has been almost single-handed the greatest missionary in this respect, and certainly the most effective one. The live polio vaccine has been, by all odds, the most effective means of control, even though evidence has been presented that it is less effective in tropical countries than it is in the temperate climate. In my opinion, this has been used as an excuse more than a scientifically important observation. It has been used as an excuse for administrative failure, for a lack of political will. It has been clearly demonstrated in those countries in the tropics where they have put on programs of sufficient intensity. And here again, Albert has held their hand and led the way. They have been able to control polio. Now, that's not to say there are not problems. The stability of the vaccine is still a problem. The availability of infrastructure is still a problem. And there has been a constant conversation back and forth between those who favor introducing vaccination programs into primary care programs as opposed to mounting specific campaigns. Well, that's very well, and we'd all like to see that. But if you don't have a good primary care structure, that's not a very effective way to approach matters. And much of the world does not, and is not going to have it in the near future. On the other hand, you can control polio in countries that do not have strong infrastructures by using the techniques that Albert has been so vocal in emphasizing around the world. Now, each country has to use its own, adapt to its own peculiar circumstances, but it can be done. There is still worry about the very infrequent case of vaccine-induced disease, and that's an argument which is being used, particularly in countries where the disease is almost totally eradicated. But for much of the world, that is a very, very small price to pay. Now, with the newer vaccines, killed vaccines, particularly with ones that are combined with DPT, if we can ever get over the P problem, namely the concerns about pertussis, this may become an effective component of the routine immunization. And it is, as we'll hear later in this program, theoretically possible to prepare killed vaccines that are more effective and are combined in a way that is now made possible by molecular biology and recombinant techniques. But if killed vaccine is to be used in much of the world, the numbers of doses have to be reduced, the cost has to be reduced, and the means of administering it is still a problem. Now, I know that Albert would like to see the disease eradicated, or the agent, and he has written on this subject. And the Pan American Health Organization has adopted an eradication program in the Western Hemisphere. Polio has some of the attributes for an eradicatable agent. There is no animal host. The vaccines are effective. We know from experience that if you immunize populations intensively enough, you can eradicate the agent from that environment. But it also has some attributes that make it difficult. I've already mentioned some of the delivery problems, the cost problems, the high proportion of non-clinical infections, the fact that other agents, and Albert has written on this, that other agents produce paralysis, and that makes surveillance difficult. And finally is the issue of political will. Are the countries involved willing and able to do what's necessary? And here's where Albert has been incredibly effective in introducing political will, which ain't easy. Well, I'd like just to introduce a couple of personal anecdotes, if I might. Albert's critical faculties had already been referred to. I became a member of the Virus Commission of the Armed Forces Epidemiologic Board, and at the first meeting I went to, I discovered that Albert had just finished his term. Well, somebody was presenting, and I kept asking questions. I happened to be quite interested in whatever was being presented. And somebody turned to me and said, "Who do you think you are, Albert Sabin?" Well, I didn't think I was Albert Sabin, but I was a little jolted at that one. I have seldom been able to emulate Albert Sabin, and I certainly have never been able to emulate him in the beard department. There he really distinguishes himself. And although we all know Albert as Albert Sabin, I suspect a good bit of the world thinks of him as Moses. Another experience that I had with Albert, it shows his nose for the experiment. When he visited us one time, and we showed him some work that was going on otitis media, where people were puncturing the eardrum and aspirating the middle ear. They were having difficulty distinguishing the agents they grew, so whether they came from the middle ear or came from the contamination of the external canal. And Albert listened to this, and he said, well, he says, why don't you just culture the external canal and then subtract? Well, they did that, and it worked fine. Here's an example of a totally different problem that he'd never dealt with before, and he immediately went right to the heart of the matter. Now, I think Tom gave the paper, the first paper given on our tissue culture experience. Albert discussed that paper. And his first remark was, I don't believe it. I tried it and it didn't work. Well, that irritated us a little bit. But after the meeting, he came up to us and he talked about it, and it was clear that he really wanted to know, and he has been a great supporter ever since. He's also a competitor. I remember when we were doing some work on antibody in breast milk, we went down to see Albert because we'd run across an abstract that he had in the literature on many years before on anti-poliomyelitic substance in breast milk. And we thought he'd be interested in our work, and sure enough he was, very interested. His paper on breast milk appeared just before ours. Well, I think we all